Hi, everyone. Thank you for joining today. We'll get started in about 5 minutes. All right, let's go ahead and get started. Hello, everyone, and thank you all for joining today. Welcome to the NOAA Omics seminar series. I'm your host, Nicole Miller, and I sit in NOAA Ocean Exploration. Omics is a suite of tools used to analyze DNA, RNA, proteins, and metabolites. It's become a large priority here at NOAA in the past few years, and we established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from this series are made available on YouTube and shared to our NOAA Omics website. There should be a link to the website in the chat now. As participants, you are muted, but feel free to type questions in the questions for staff box throughout the presentation. And at the end, I will read the questions for the presenter to answer. So with that, our presentation today is titled eDNA Monitoring in the National Estuarine Research Reserves, Lessons on Standardization, Communication, and Implementation by Dr. Alison Watts. Dr. Alison Watts is a research faculty at the University of New Hampshire Department of Civil and Environmental Engineering. Dr. Watts' main research interests include working with regulatory agencies and resource managers to assess and remediate water resource impacts. She has worked extensively on watershed planning and restoration, green infrastructure implementation, and water quality assessment. Her current work is focused on the use of molecular methods, primarily eDNA, and includes early detection of invasive species, water quality indicators, impacts of aquaculture, and baseline assessments in coastal systems. Thank you, Dr. Watts, for presenting today, and I will now pass it off to you. I can see your slides clearly.
Awesome. Thank you so much, Nicole. And thank you to all of you lovely faceless people that I can't see out there, but thank you for showing up and being interested in this work. So I'm going to talk about a project that I was just doing dates and realized we have actually been working on this for nearly 3 years. We started early about 3 years ago. And what we've been doing is working with the National Estuarine Research Reserves and trying to understand how eDNA fits into a water quality monitoring program. So I am having trouble forwarding my slides. Oh, there we go. So the National Estuarine Research Reserves, NERS, probably many of you have at least heard of them. Some of you may be working at them. It's a network of sites around the country in coastal locations all across the coastal areas, including some in the Great Lakes. It's a partnership between NOAA and agencies within each of those states. And they have many fine characteristics, but one of the things that they do is they do a standardized water quality monitoring program. So every site goes out monthly and collects the same parameters using the same methods. So that's sort of a standard sort of format for collecting water quality data. In this project, we can work with all 30 of them. So we picked 10 sites to work with. Those are the yellow stars on the map here. And we tried to work with sites that represented a variety of coasts, different types of conditions, urban, pretty rural, Gulf Coast, all the way over to Hawaii. The science coordinators for each of the reserves are sort of listed there. So there's a science coordinator at each reserve who is our point person for the work that we're doing there. And then each of those people has several people who work with them doing the actual sample collection and helping us with the data interpretation. So there's really is about 30 people involved in this project, and they're all just been completely awesome. I thank them so much for showing up and for sharing their knowledge with us and for going out in all kinds of weather and taking samples. And so our question was, they're already taking these standardized water quality samples. What happens if we just drop eDNA onto that existing program? So I'm sure most of you have heard many eDNA talks, and they tend to be around a particular question. Is the species present in the system, or can we measure seasonal change in these streams, something like that? We're not asking a specific question about the conditions at each site. The question we're asking is, how do we make eDNA useful in this context? So sort of the methodology here, all of the sample analysis is done here at the University of New Hampshire. We put together sample kits, basically jars of preservative, filters. We mail all of that off to each of our 10 participating sites. They do the sampling. We have a protocol for doing that sampling. They collect the samples mostly on boats, some of them from bridges or roads, but mostly from boats. Then they take them back to the lab. They filter them in place. One of the things about a program that already does water quality monitoring is they probably have to filter their nutrient samples anyway. So nitrogen needs to be sampled pretty much immediately after collection. So they tend to have these filter manifolds. They have the ability to either acid wash or bleach equipment. So they're pretty well set up for doing this kind of filtering. We took triplicate 1 liter samples at each site along with a blank. Each NER sampled 4 to 5 sites, and the sites were all part of their existing water quality network, which they have been sampling for years and in many cases actually decades. So it's a very long-term data set. We sampled quarterly over the course of 2 years. Those samples were then shipped back to UNH. We do the extraction. We ran them for 12S for looking for fish, my fish primer, and 18S looking for eukaryotes. There's about 1,200 samples. So there is a ton of data here. In terms of sort of the methods, sampling in estuaries has some particular challenges, and anyone who's been out there knows this. They're often very turbid systems. So this picture here, this little stream, that's one of the sampling sites in San Francisco. It's a very, very turbid, marshy stream. And that turbidity translates to clogged filters. So this is actually a filter from Great Bay. That's one of our not so turbid sites, believe it or not. So there are often multiple filters to get a liter of water through, and it's pretty time consuming to do that. Because of those time constraints, they really can't filter in the boat. They have to bring them back. They just don't have time to sit there and filter all of these samples and sit there because, amongst other things, the tide is going down. So they try to sample, many of these sites, their samples sort of at the same tide. This, I think, is GTM near in Florida. That looks like an oyster reef. You don't want the tide going down and leaving you on the oyster reef. So there are a bunch of constraints there. There's a lot of turbidity, which affects your filtering time. There's a lot of inhibition because there's so much organic material, and there's these time constraints about when you can do the filtering. So the methods that we recommend are really sort of really aimed at that particular site characteristic. So these might not be the methods you will want to use if you're working on a clear stream somewhere inland. If you're working in a turbid area, these methods seem to work pretty well. Our protocols are all on our website, so estuarydna.org. I think if you just search UNH eDNA, you'll probably get there. So the field protocols, all those little videos are on that website. Website also has the lab protocols. The lab protocols are also published. After our first year of sampling, we realized we just were not getting the fish species that we expected to see. So we went back to the lab and did some more optimizing. And nothing in this optimization is particularly new or unusual. It's all the things you know you want to do, like doing inhibition removal, doing PCR replicates. We realized we needed to do all of them. And we needed to be very specific about what we were looking for. So we optimized our protocol and went back, and we had much better results with that cleaner protocol. So thinking about a standardized eDNA assessment and monitoring program, what if eDNA were just another parameter that we went out and monitored? What if it weren't this researchy thing that we only use under certain circumstances? So say you went out and collected these samples and you have all of this data. The first question is, can managers use it? How's it useful? What are we going to do with all of this information? What does eDNA add to a water quality monitoring program? So you're out there, you're collecting the water quality parameters. What does the eDNA add to the knowledge? If you're working in a network, so one of the great things about NERs is that they're all over the country, well, coastal parts of the country, and they're all doing the same thing. So what if you take the standardized program and we drop it into a network that's in a lot of different locations? What kind of additional information does the network give us? And what does co-locating water quality add? So all of these sites sample monthly for water quality, pH. Well, to be honest, the cold ones up here in New Hampshire, they actually pull this stuff in the middle of the winter because it freezes, but most of them sample monthly. pH, dissolved oxygen, temperature, stuff like that. This picture is actually Apalachicola. They're pulling the nutrient samples, I think, I hope, since they're not wearing gloves. I don't think it's the eDNA. And that behind them is their water quality tower. So that's got real-time sondes that sit there collecting continuous data. So we can co-locate the eDNA, the nutrient, and this other water quality monitoring. Most of these sites do not have consistent biology monitoring. So for instance, many of them do fish surveys, but they're not using the same method. Apalachicola does trawling. In Great Bay, they do seining. They don't necessarily do it in coordination with their water quality monitoring. So although they have a lot of information about their systems, they don't collect that same information in the same way. And one of the things that we were hoping is that the eDNA would give them a standardized and consistent set of biological data that they could compare or use along with this water quality data. So let's start with the first and most important one, right? How do managers going to be able to use this? So even in this study, which is by no means all of the NERs, there's a lot of information. So 3,000 different eukaryote species, hundreds of fish, all of this stuff about ASVs, and you start talking to managers about ASVs, you get this like, really? Why are you telling me about this? So one of the things we're really working on is how do we sort of compartmentalize this information into pieces that are useful and interesting to managers. And one of the easiest things usually is fish. So fish are, there's a lot less fish than there are eukaryotes when you look at the whole sequence set. And pretty much everybody's interested in the fish in their system. Some of them have information about those fish and some of them don't. So when we look at this full data set, we got on the order of 320 total fish species across all of the sites. And when we take that and we ask people, is this pretty much what you'd expect to see? Generally, we are capturing the expected species. And I'm going to go in and I'm going to talk a little about the 30, the ish part on the 320 and what we mean by expected. So if you're working with eDNA, most of you probably know that what happens is you have a sequence, which is basically a string of letters, comes off the sequencer, and there's a process by which you try and match that set of letters to a species in a database. And in that process, sometimes you get matches to species that are very peculiar. Sometimes you don't get an exact match to a known species. So that automated process is great for probably 90% of your species. And then there are some that you really may give you results that look very peculiar. So one of the things we really try and be careful about is not to give managers lists of fish that they know are not in their system, because once you've made that mistake, you've really lost a lot of trust in this process. So what we've sort of come to, and there's actually a whole MBON working group right now that's working on sort of putting together guidelines and a process for this, but this is what we came up for in this particular project. We start with the initial species that comes out of the bioinformatics pipeline. So that's the first column. We do a check on that species from just a database like FishBase, and we just say, is this even reasonable to be present here? And sometimes we correct that based on whether or not it's reasonable to be correct there. And then we send that database, this table, off to the managers at the location, and we say, double check this. Tell us what you think is reasonable. So this looks like Jacques Cousteau NER. And what they've done is they've gone through in this column and they said, yep, it's common, it's documented, it's not really, we haven't seen it, but it's not unlikely to be here. And then some of them, like the convex ciliate, that's an aquarium fish. We flagged it as this is probably not likely to be here. They agree. So that's not likely to be there. We're not quite sure whether it's contamination or how it got in there, but we flag it as a species that's unlikely to be there. So if we walk through the process here, so let's look at this line here. It's Microptus. This came up originally as a Guadalupe bass, which is only found in Texas. When we take that over to the JC managers, they say no, but we do get mud sunfish. And when we do a blast, we see that we match mud sunfish with the same level of certainty. So we say, okay, Although the automated process gave us this fish, we are going to decide to call it mud sunfish in this database. And we have a set of flags. So 100% match is 100% match in the database, and it's likely to be there. That's a 1. That's most of our stuff, which is good. Sometimes we get things like this amatoides. It's 100% match to a couple of different species that are likely to be present. We can't tell which one it is, so we're not going to make that call. We're just going to say it's something in that species, and we're going to flag it with a 2. 3, so that's our mud sunfish example. It matches multiple species, but only one is likely to be there. So we're going to make the assumption that it is that species, but we're going to flag it just in case somebody says, wait, who knows, but this other version of that other subspecies is coming up from the south or something. And then 4, we don't want to take these 4s out because maybe there's a reason why these very unusual species are present, but we want to make it clear that we understand that they're probably not living in the system. So all of the fish data that you're going to be looking at has been very carefully reviewed by the managers in the system. And yes, this is a really time-consuming process. So let's take a look at an example. Let's look at Apalachicola Bay. This is in the Florida Panhandle. These are 4 sites that they're sampling. And so this is 2 years of data, 4 quarters. One of the questions that is asked a lot is, how does eDNA compare to other methods? I'm out there doing this trawling, can I just stop trawling? Can I use eDNA instead? Does it give me the same species? Is it better? Is it worse? Most of these sites don't have side-by-side data, so we can't do that comparison in a lot of sites. Apalachicola happens to do trawl sampling at pretty much the same sites, not necessarily the same day, but very close to the time when they do their water quality sampling. So this is pretty close to side-by-side comparisons. If we look at the 2 data sets, the trawl and the eDNA, what we see is that we have eDNA only, about 83 species, trawl only 63, and relatively little overlap between the 2 methods. So both methods are giving us like 128 species. There's a little bit of mismatch here because some of these eDNA, we can only get to the species level and we can't match them to the trawl. So there's probably actually a bit more overlap than we're able to show here. And when I first saw this, I was really floored by it. Like, why is this so different? We looked at some of our other projects, other sites. So in Great Bay, we also have comparative data. We have more overlap, but it's not complete. San Francisco, where we have fewer data sets to compare to, we have very little overlap. But the thing is that both of these methods are only capturing a portion of the species that we expect are present. So when I went back and looked at the list of species detected in the trawl data over the last 10 years, there's more than 300 fish species there. So the trawl doesn't get them all, the eDNA doesn't get them all, but if you put them together, you're getting closer to that full picture. And as I talk to people more and more, I'm hearing that this is kind of what people are seeing, particularly in coastal areas where things are sort of complicated and species may be in little niches sitting in some areas and not in others. And to me, this is not one's method is better than the other. These methods, it's like doing twice as much sampling to some extent. You're doing 2 methods, you're getting a lot more information. It's possible, it's probable, in fact, that the eDNA is to some extent slanted in some way and the trawl is slanted one way. The people at Apalachicola, we're going to try and dive into this data set a little bit deeper and see if there's a reason why some species, for instance, might not be, larger fish might be jumping out of trawls or something. Is there a pattern to what is being detected in the different methods? But the message there is they're all just snapshots. None of them are going to give you the full picture. If we kept doing this year after year after year, these circles would start to converge. But if we're just looking at a fairly, 8 sets of samples, we're not going to get the same results and we shouldn't expect to. And we need to be really clear about that. When people ask us, do we get the same results with these methods? Probably not, and probably because they're both incomplete. The other question we're asked a lot, of course, is can we measure abundance with eDNA? And there are various smart people working on this, looking at things like using standards. We didn't do that. We're not doing any of that sort of more method work. But what we did do is we just take the number of times a species is detected. So we're not looking at counts, we're just looking at yes, no, detected, not detected. And because we're doing triplicate samples, we actually have a fair number of samples we can do this with. And we just say, what is the species that is most commonly detected along the eastern coast, the northeastern coast? So this is New Hampshire, New Jersey, and Maine over the course of a year. And then we show this to managers. I took this to a local fisheries conference, and I show this graph and I say, is this reasonable? Is this kind of reflects the species that you think are most prevalent in this area? And what we get is yes, but. So generally speaking, this is a pretty good qualitative measure of what's present. We are missing some key species. When we look at them in more detail, they just are not in the database for this primer. So as the databases get updated, those species are going to start to pop up in here. The databases misname things in weird ways sometimes. So I am often asked, what the heck is it's in here somewhere. What's the bluegill? What's a blue herring? And sometimes it's just, it's blueback shad. So it is, blueback shad is blueback herring. And the database calls it shad. We can fix that, but those little errors also really kind of throw people off when they're looking at this. Okay, diving into fish a little bit more. This is Guana palmatomazina, and I can't pronounce this, so I'm just going to go with GTM. This is also in Florida, but it's on the other side. So this is the Atlantic side. GTM does not have fish monitoring directly in their system. There's monitoring that happens sort of in the region. So they have a general sense of what species are present, but they don't know exactly what's at their sampling sites. So for them, the fish data is kind of new and fun. This is broken out by family. There's 120 species that we detected in the system. I can't find enough colors in the rainbow. So just trying to do this by family so we can get a sense of how we can snapshot this information. The gray here is mullet, and it's everywhere. It's in all of the samples. And when we ask them, they're like, yep, there's a lot of mullet around. So they may not be saying anything, but they say, the fishermen say there's mullet. Everybody who lives there knows that there's just a ton of mullet. So that's nice. We can look at this in a little bit more detail. So these are their 4 sampling sites. They're pretty much sampling inside the barrier islands. This creek here, Pellissier Creek, is actually fairly far up. So that's almost freshwater, semi-tidal but semi-fresh. And we can see on the graph that particular site has a whole lot of freshwater species. So that's mostly bass and sunfish. We don't really see much of that anywhere else. So that's a reflection of where that is located in the landscape. We also detected American eel just a tiny bit at every site. They were kind of happy about that because they suspected it was there, but they've never gone out and done the monitoring to confirm that it's there. So it was nice for them to know that that species that is important to them is present in the system. So carrying on with the fish species and thinking about the value of a network. So we can take those sites along the East Coast, Maine, New Jersey, and Florida, and we can do a heat map of what species are detected. And this was something like, I can't remember, 160 species, and we can't list all the names, so I didn't. The darkness is, again, the number of times a species was detected. So very dark blue, detected a lot of times, is plain yellow, not detected. And you can see that in Maine, there's a bunch of species that are detected, and then we start seeing those drop off as we go into Florida, and the inverse as we go into Florida. There are species that are present that are not detected in these more northern sites. And there's nothing surprising about that, right? We all know that different fish like different conditions, and the fish in Florida are different from the fish in Maine. But this is a way you can get at that using this sort of standardized eDNA method and do this direct comparison without worrying about whether or not it's because you're using a different net size, or one of you is doing larval toes and one of you is doing adult fish. So you can do that comparison. Because we have water quality data, we can, for instance, look at fish by temperature. So just bidding 10 degrees Celsius, and yes, this is Florida, some poor little very hot stream. Again, not surprising, but the fish like it better when the temperature is within sort of the mid-range. So we can use that to sort of see how species correlate to temperature and other water quality parameters. The other thing we can start to pull out of this, so just stopping on fish, enough on the fish, moving on to other species that people have expressed interest in. Everybody's interested in a picture of an otter. So although we are running our analyses for fish, the MyFish primer that we're using also picks up mammals. So this is, if you like, it's bycatch from our analysis. We do pick up some of these endangered species, and that's nice for managers to be able to say that and to be able to sort of convey that this method is also helping them to understand some of those issues with endangered species. We didn't pick up a lot of invasive species, but that's also potentially possible with this method. So we also did this analysis for 18S, for eukaryote species generally. If we look at that data, here is all of the sites grouped together. So that's all the samples from all of the sites. Again, way, way, way too many species and subspecies to do individually. So we brought this up to phylum. So the colors of the different phylum. This is all of the sites together. And then each of the 10 sites separately, but within each site, we've been together all of the quarters and all of the locations. So that's a summary of what's there. What's interesting to me really is that these kind of look the same, at least at this level. So although there's certainly variations, there's a general sense that there's a lot of these orange and sort of yellowy species. There's a little bit of this purple at most sites, but not all of them. The exception is Mission Aransas. This is in Texas. So this is Texas coast. And that one does look different. And we are doing more analysis on this, but just visually. So that one does look a little bit different. This purple color here is this well-known species not available, but we can dive into it. If I look at it a little bit more closely and look at the sequences, a lot of these are dinoflagellates. And why? I don't know. Don't know if that's just something we were catching there. That's a very turbid site that has a lot of algae and other growth. So maybe that's a reflection of that. If we look at this a little bit more detailed, one of the other questions that we feel that managers seem to be very interested in is metrics of change. How do we tell when things are changing, whether it's natural change, whether it's seasonal change, or whether it's because some intervention or some remediation has happened? How do we know whether the biology is changing? So the 18S is a really great way of looking at that. We're going back into the GTM site. These are the 4 sampling locations, the FM, PC, PISS, and you can see that there are differences between the sites. So this is all 4 quarters bin together for each of those sites. And then we can break those out by site, by quarter. So PC, this is our little friend here, up that freshwater creek. And you can see that there's really significant changes. So this is winter, summer, fall. It was November, August, sorry, February, August, November. And there's a pretty significant changes happening there. If we look at that in more detail, it's hard to do because the colors all kind of run together, but this winter, there's a lot of arthropods floating around in here. As we move into summer, Silphora start blooming or whatever it is they're doing. I'm not a biologist. And then in the fall, Rhodoptera start moving in. And because we don't have long-term data, we don't know if this is something that happens every year, but we can look at this and we can say, okay, this is what happened this year. And if you're interested in change, you could come back in a couple of years and you can see whether or not you're still seeing the same pattern, whether this is something that happens every year. And you can compare particular species and see if they come and go. So this is a way of getting a snapshot of your system at that particular time that you can compare to what happens to it later. In terms of questions that people are interested in, harmful algae are always kind of high on the list. So people are really interested in whether or not there are harmful algae present at their sites. We were specifically asked to look into Pseudonychia, which is a nasty neurotoxin, which has really significant effects on wildlife. It's known to be occurring along the West Coast. So these blue and sort of greeny blue, that's South Slough and Padilla Bay in Washington and Oregon. We were actually asked to do this by GTM, which is orange, because they were also doing a tow analysis and they wanted to know, they were seeing it and they were surprised to see it. And they wanted to know if we were picking it up in our analyses too. And the answer is yes. And for some reason, it seems to be blooming there more in February, whereas in the West Coast sites, it's blooming more in the summer, and that's probably a temperature thing. What's also interesting about this data, though, is the green here is wells in Maine, and it's pretty chilly up there, and we weren't really expecting to see it. It turns out it is known to be increasing, so whether that's the warming temperatures in the Gulf of Maine or what exactly is happening in there, they are starting to increase, and we are picking that up in this data set. I can't say that's nice exactly, but I can say it's exciting or it's interesting. And then because we have the co-located water quality, right, we can take those pseudonychia, and one of the things we can do with the molecular method is we can get down to species for the pseudonychia. Apparently this is hard to do with a microscope, so it's nice for them to be able to see that we can start to differentiate the species because not all of them are toxic. So pseudonychia as a class kind of is one thing, but to be able to get down and determine whether or not you've got the toxic species present or not is really helpful. We can map it against various water quality parameters. The ones that really stood out was salinity. So it's definitely associated with higher salinity sites, and we can also start to look at whether particular species, like the toxic species, are more associated with these higher salinity sites. Speaking of toxic algae, so the other thing we're getting into is just the general exploration of the data set. We're looking for patterns, we're looking for things that change. So if you look at the entire data set, and Jeff Miller, who's doing this analysis, is on this call, so you can interrogate him later because this is not an analysis I did. The thing that is ranked as the highest temporal signal, the highest change, is this chloromorium, which is again another form of toxic algae, highly elevated in August in these are the Gulf sites. So GTM and Apalachicola Bay are in Florida, Mission Aransas is in Texas. So in the Gulf sites, in August, there's this sudden bloom of this thing, and then it goes away again. That's just a piece of analysis that's sort of come out. We'll be following up on that with those sites, but we can look for other things. Like I think one of the other things that came out of this was mussels. So there's a big flush of mussel at one particular point in time, possibly coinciding with spawning. So we might be able to start picking up spawning events. If we co-locate it again with the water quality, maybe we can use the water quality to start to predict when these events may be happening. This is kind of where what we're working on now is understanding the relationship between community and location and community and water chemistry. So these are principal components analyses. The points each represent a sample, and points that are together generally have the same species in them. So red is generally samples that were collected in the southern sites, and blue is samples collected in the northern sites, and the community in those sort of north-south divide is distinctly different. To be honest, I thought we would be seeing a pretty big difference between things like oceans, like the Atlantic versus the Pacific, and we're actually not. And I think it's partly because the data set is so complicated and there's so many different variables at play here, it's hard to pick them out. One of the things that is a really big driver is salinity, and that's not surprising, right? Because we know that fresh versus saline is one of the big dividers in water biology. But again, here, so these red sites, these are all in the higher, sorry, that's lower saline, and higher salinity sites, but they're very distinctly different communities. And that signal is so strong that we have to sort of think of ways of seeing signals that are a little bit subordinate to that and picking them out of here. And that's really what we're working on now. And I'd say anybody who has bright ideas, feel free to chime in and tell us about it. So this question of what does eDNA add to water monitoring? None of the things that we're picking out are things that were necessarily completely unknown at these sites. They're not wildly new, they're not wildly different. The thing that is different about them is we're getting it all out of one sample. So that, to me, that is the game changer. That's the place where this becomes not just another method, but an overarching tool to really understand your system. We're getting the fish, we're getting endangered species, general metrics of change, baseline biology, HABs, all of that out of one sample. And these samples, frankly, are a pain in the neck to collect. The filtering takes a long time, but the amount of information we get out of them is really extraordinary. So I've talked a lot about what we are doing and what we can do to, you know, how we think we're presenting this information. I can't tell who's on this call, but I'm hoping that there are some managers, resource managers or fisheries people who are interested in eDNA. We need your feedback. So we need for this project, but also anyone who's doing this type of work, we really need the users to get back to us and say, that is super useful. We can use that. That is probably great research, but I don't know what the heck to do with it. I can't do anything with it. And we need your honest opinion on whether those species are actually present, whether you've completely lost trust in what we're doing and you're going to say, you're telling me those things are there. We know they're not. I just don't want to use this method. We need that feedback, and then we can go back and we can look at our methods. We can adjust them. We can try and make them more precise. You do a reality check, but it does take a time commitment on the part of the resource managers. So the people at the NERS who are working on this, I am, as I said, really grateful because they put the time in. And they still have to put up with me. I'm still calling them up all the time and saying, oh, is this there? Does this matter to you? What would you do with this data? And that's a commitment on both sides to keep that conversation going, and it's super important. So that's just a really brief overview of what we're finding in this data set. We do have a website, our draft data under the data and results section. If you're interested in, if you go there, there are the tables with the data. We don't have any of these analyses up yet, but we will put them up as we get through less drafty versions. Our protocols are all up there if you're interested in how we do the actual sampling. These lovely people, can't remember where these ones are, maybe Jacques Cousteau, are sampling. And of course, they're having a great time because everybody has a great time when they're doing eDNA sampling, right? So check that out. And if you have any questions about that, get back to me. And then before we move into general questions about the project, I do want to say that we have some funding available that supports our time to help agencies or managers who are interested in using eDNA but have questions. So if you're interested, but you're not sure if eDNA is the right thing for the question that you're trying to ask, if you want to know what methods might be appropriate, if you want some help designing a sampling plan, we can do that for you. We can help walk you through that at no cost to you, except your time, and I will make you put in the time. So if you have something that you're interested in, or you're just interested in knowing more about this, send me an email, or I think you can probably drop a note in the questions. Nicole said that we will be able to save the questions so I can get back to them later. So if that's something anyone's interested in, please let me know. And then questions. So I'm interested in questions anyone has about the project, but I'm also interested in any suggestions you all might have about how we could use this data. So other ways, other things that we should be looking for. It's so big that we sort of wade into it and we say, oh, okay. It would be helpful if there were directions, if there were particular things that people thought we should be looking for or experiences you all have had. We would love to hear them. And Nicole says the best way to do that is in the questions for staff box, I think.
Correct, yep.
Okay, thank you for your time and attention.
Thank you, Alison, for the great and exciting presentation today. So many exciting opportunities and ways that we can look at our estuaries and our ecosystems through the lens of eDNA. So thank you very much for that. At this point, we'll move into questions. So if you have any questions for Alison, please feel free to put them in the questions for staff box, as well as any comments or feedback on where Alison should direct some of her future efforts in analyzing this data. So we had two questions come in, and we'll get started there. The first two are from Julie, who asks, do the protocols detect mollusks? And I think you touched on that a little bit, that there was a point in this study where mollusks were detected. And then the second question is, could the eDNA detected in the estuary have originated from a freshwater stream versus the marine ecosystem? And are there any hydrology measurements that may inform what we're seeing in these samples? So we'll start with the first, with the mollusks, and then move into where hydrology of freshwater versus marine may impact this data.
Okay. Yeah, so the mollusks, yes, we do pick up mollusks and invertebrates in that 18S analysis. It's not designed, if you were specifically interested in mollusks, you would probably want to use a primer that is more specific to mollusks. So the 18S is really good for broad eukaryotes. It's not so good for getting down to particular species. So in some cases we can, but a lot of cases we might just say, yeah, it's a mollusk, it's an arthropod. So if you're interested in a specific species, you might, like we did fish, to focus in on the fish, and then the 18S to look broadly. You might want to do an 18S and then something that's more specific to mollusks. And then, yes, absolutely, we do indeed pick up freshwater species that are not living in that system. So the freshwater that I showed you at the GTM near, they might have been living there. I'm not quite sure how far up that stream. I could tell by looking at the salinity, but we definitely do get DNA from species that are living up the river that flow down. And that's, you know, maybe that's a confusing piece, or you can think of it as a bonus because you're also getting information about species that are further up in the watershed than where you're sampling. But absolutely, you get transport from species upstream, you can get transport from processing plants. There are a bunch of ways in which you can, I wouldn't call it a false positive because the DNA is actually there, but it is not from an organism that's living in the system.
Thanks. Thanks, Alison. Our next question is from Rebecca, who asks, could the high abundance in dinoflagellates you're seeing be due to the huge size of the dinoflagellate genome, not necessarily due to true abundance?
I mean, in that particular case, what I was looking at, and honestly, I'm not a genomics, I'm not going to answer that in detail. Probably other people on this call could, but in the case of that particular comparison, no, because we were looking at relative change. So we were looking at the same thing. We were just seeing that there was a lot more of it at that particular site.
Awesome, thank you. Our next question is from Carol, who asks, a very interesting talk, but what are the qualification controls for the relative representation of these taxa in these comparisons? How are you controlling the marker or primer bias among your detected data?
Yeah, so that's a great question, and we aren't. So people are. When we started this project 3 years ago, I think that was something people were just starting to dabble in, and people are definitely doing more of that now. But that is part of the reason why I'm looking more at number of times detected than abundance of reads, because you're absolutely right. Reads are, it's a nonlinear amplification, and if you don't have some kind of standard, you don't know whether it was just really, really happy with that primer and you have primer bias. It's not really bias, but there are gaps, so different primers are not, the fish primer that we're using doesn't pick up all of the species that we know are in the system, and that's more of a gap in what that primer can detect.
Thanks, Alison. Our next question is from Eric, who asks, or states, I can understand why some species might show up in the eDNA and not in the trawl, but why would species be caught in the trawl but not necessarily show up in the eDNA samples?
Yeah, so a couple of reasons. One is that potential primer bias. So there are species that just aren't necessarily as good for that primer, but definitely not 63 of them. They're not taken at exactly the same time. So we are not dipping the sample while the nets are in the water. They're coming out the day after. They're not in exactly the same location. So it's not exactly, they're not exactly paired samples. And I think that's genuine variation. If you look at, if you took, and we have done this, although not there, if you take a bunch of trawl samples 3 days apart, they're not going to give you the same species either. Whether there's, I think that there are probably some species that are more suited to one method than the other, and that is something we'll be looking at. But I think a lot of what we're just seeing is true patchiness in the data, in the actual species presence.
Mm-hmm. Thanks, Alison. This question is by Jonas, who asks, I work with local watersheds in the San Francisco Bay, and there has been some talk of eDNA monitoring. but Alison, do you know if there's any ongoing monitoring within that area, in that estuary?
Yes, I know there is. I don't know the details. I will actually be out there in April, so I could ask, but I will know more then. But yeah, I know, I believe that the San Francisco Bay Estuary Institute is doing some. I know several of the universities, San Francisco State, Davis University, I know that they have programs in the estuary. There was another program. There are several programs that are doing eDNA. They tend to be focused on a specific thing like delta smelt or invasive species, not so much the broad just what if monitoring, but there's a fair amount going on there.
Thank you for that. Lots of great questions coming in, guys. Thank you so much. This one is from Rebecca, who asks, in addition to presence absence of HAB species, is there a way for the DNA analysis to measure toxin production for HABs? Sometimes they're present but not actually producing toxic compounds. And maybe that can be explored within the water quality measurements taken as well.
Yeah, so that's a great question. The method that we're using, that's a no, because all we're doing is we're just looking at the section of the DNA that says, yes, I'm an algae, no, I'm not. There are methods for looking at gene expression that would tell you more about that, maybe combined with chemical analysis of the toxin in the water. So the method that we are using, no, I don't think that the samples we've collected could be pulled out of the freezer and used for that either, because I don't think they were preserved correctly for that particular question, but there are people who are using molecular methods to answer that.
Mm-hmm. Thank you, Alison. Our next question is from Jeff, who says, great talk. For those planning eDNA experiments, is there a general cost for how much it costs to collect and sequence and then analyze a sample?
So I usually use Jonah Ventures, which is one of the relatively few commercial, I mean, we do our stuff in-house, but in terms of costing, I should look this up. I think that they are sort of on the order of $50 to $60 for a single species, and maybe on the order of $75 to $120 for the metabarcoding multiple species. If you work with a government or an agency lab, they can often do it for less because they have the equipment on hand, but that's sort of the price range for the commercial, I think.
Thank you. Thanks for that, Alison. This is a follow-up question by Lauren on the freshwater/marine hydrology. How confident are the researchers that eDNA results indicate presence of a species on the site of interest versus in a nearby ocean or the estuary more broadly? So I think that goes into a little bit of the scale of eDNA, which is still in question and determination. But Alison?
Yeah, so it's a great question. It sort of relates to the earlier one about transport. And there's a lot of study, a lot of work going on how eDNA transports. And the answer is kind of it's different in different systems. So you really have to be careful about that reality check. You have to make sure you understand whether you always have to be open to the possibility that it came from somewhere else. And I don't really see a way around that. Generally, if it turns up over and over, it's probably more likely to be there. But if you have a source that's nearby, that source is going to be leaking that DNA into the system. If you have an open ocean nearby, we don't see ocean species generally within our estuaries. We don't see the freshwater species much beyond the mouth of the rivers. So we know it's transported, but it doesn't seem to be, it's not like you sample in the middle of the estuary and it's all full of freshwater species. It's very much co-located with the freshwater that's coming down.
Thanks. Thank you, Alison. Okay, our next question is from Michelle. I'm going to paraphrase as best as I can. Right, there are many uncertainties when understanding the applications of eDNA. And Michelle asks, with that in mind, what resources are you aware of that can help managers understand these considerations? Some are, thanks Michelle for noting, some are available under the resources tab on the NOAA Omics landing page, linked in the chat for those that are in attendance. but there's no page right on particularly estuary DNA. So would you recommend any other resources for managers that want to bring these types of methods into their research programs or monitoring programs?
So estuary specific, I don't know of anything off the bat. So there is the NOAA eDNA page. There is the eDNA Collaborative. I think if you just search eDNA Collaborative, they have a lot of great resources on their site, including some nice eDNA fact sheet things. We have on our website, we have the methods that we use and that are sort of estuary specific. I'm happy to talk to you about what we feel works in estuaries and what is different about that. But I don't know of any like website for estuaries. I guess ours would be the closest, and I should be putting more stuff up on it. Good point.
Awesome, thank you. Okay, so we'll have one more final question today by Peter, who asks, is there a system for archiving samples so that DNA can be used or collected in the future for historic preservation and or backlogging?
Yeah, so another great question. So the sequences themselves go up on the public databases. So that's after it's been run. The samples, if you mean the physical samples, are sitting in our freezer, and in theory, we keep them, and in theory, they're available. But the reality is we have to find them. We have to have someone who can sort of dig them out. There has been a lot of conversation about places like the Smithsonian possibly becoming a holder of those samples, some kind of archival database. I don't quite know where that is, probably not happening right at the moment, but it's definitely a need that is recognized and that people are thinking about.
Awesome. Well, with that, Dr. Watts, thank you so much for your great presentation today. And don't forget to post any other feedback that you might have. We'll stay on just a minute longer to post any feedback you may have to reach out to Alison after this seminar. With that as well, thank you all so much for joining us today. Our next seminar series will be Wednesday, April 23rd by Diana Batester on a practical approach to integrating eDNA into stock assessment in fisheries management advice. So a great follow-up to this month's seminar series. See you all next time. And once again, thank you, Dr. Watts, for joining today.
Awesome. Thank you all. Bye. All right, Nicole, do I log off?
Yep, I'm ending it now. Thanks, everybody.
Okay.